Hello everyone, welcome to my class. So today we will talk about polysaccharides. So what are polysaccharides? These are the polymers of monosaccharides. Means simply the carbohydrates which on hydrolysis produces many number of monosaccharide units. For example, starch, glycogen, cellulose. So let us talk about the first one, that is starch. So what is starch? Starch is a polymer of alpha D glucose and a major reserve food in plants. It turns blue with iodine. Actually, starch is a mixture of two components. One is water soluble component and another one is water insoluble component. Now water soluble component is called amylose and it is just 20% of the molecule. Amylose is an unbranched molecule which is water soluble part of the starch, whereas amylopectin is 80% of the molecule or 80% of the composition, and it is a branched and water insoluble part of the polymer. Sources of starch are potatoes, wheat, rice, maize. Coming to the structure of amylose. When hydrolyzed, amylose gives D-glucose units. D-glucose units. And on the basis of certain investigations, we can say that amylose consists of long chains of several alpha D plus glucose units. Means it's an isomer, alpha isomer by nature. Now in the chain, C1 atom of one glucose unit is linked to C4 atom of the other unit with the help of glycosidic linkage, and that linkage is C1C4 glycosidic linkage. C1C4 glycosidic linkage. Now coming to amylopectin. Amylopectin is again the alpha D glucose units which are linked together not only by C1C4 glycosidic linkage, but also they are further connected to each other as C1C6 glycosidic linkage. Means C1 of one terminal glucose molecule present in one chain will be connected to the C6 atom of another glucose molecule chain present at the terminal position via C1C6 glycosidic linkage. This is the complete structure of starch. Starch having two components, amylose and amylopectin, having two types of linkages, C1C4 as well as C1C6 glycosidic linkage, and C1C6 is responsible for the branched structure of the starch. Now the next one is cellulose. So what is cellulose? Again the formula of cellulose is C6H10O5N, means it's a polysaccharide and quite useful to us. Now what is the structure of this polysaccharide cellulose? Cellulose upon hydrolysis gives only D-glucose, means it gives beta D-glucose units, beta glucose. This means that only D-glucose units are present in cellulose, but unlike starch, these are not alpha D-glucose molecules, these are beta D-glucose molecules, means beta anomer of glucose molecules. Now, the next thing is, it's a linear chain of beta D plus glucose molecules and they are held by C1C4 glycosidic linkage. Means in case of cellulose, we have to draw n number of beta D glucose units. We have to connect them via C1C4 glycosidic linkage. So that will give us the structure of cellulose. Now the chief source of cellulose are wood. Wood contain 50% of cellulose. Cotton contains 90% of cellulose. So this is the structure of cellulose having beta C1C4 glycosidic linkages. Now several materials are obtained from cellulose. Mercerized cotton cellulose treated with concentrated sodium hydroxide solution acquires silky luster. Gun cotton, it is completely nitrated cellulose, highly explosive in nature and is quite useful in the manufacturing of smokeless gunpowder called blasting gelatin. Number 3. Cellulose acetate. It is used for making acetate rayon and motion picture films. Number 4. Cellulose xanthate. It is obtained by treating cellulose with sodium hydroxide and carbon disulfide and is a basic material for viscous rayon. Next is glycogen. Glycogen, again the formula of the glycogen is C6H10O5N, means again it is a polysaccharide. Now it is also called animal starch and its structure is similar to amylopectin, means it is a branched chain polymer of alpha D glucose units in which the chain is formed by C1C4 glycosidic linkage, whereas branching occurs by the formation of C1C6 glycosidic linkage. But there is a difference. The one main difference between glycogen and amylopectin is the length of the chain. In amylopectin, the chain consists of 20 to 25 alpha D glucose molecules, whereas in the glycogen there are 10 to 14 molecules of alpha D glucose present. Another way, glycogen is more branched than amylopectin, and in human body it is mainly present in liver, muscles and in brain. Glycogen gets converted into glucose when our body needs it, and it is being done in the presence of certain enzymes.